Hello and welcome back to my channel. This is Why the Fact Not. Coming to all of you with a brand new video. Scientists identify the cause of Alzheimer's progression in the brain for the first time. Researchers have used human data to quantify the speed of different processes that lead to Alzheimer's disease and found that it develops in a very different way than previously thought. Their results could have important implications for the development of potential treatments. This research shows the value of working with human data instead of imperfect animal models. International team, led by the University of Cambridge, found that instead of starting from a single point in the brain and initiating a chain reaction which led to the death of brain cells, Alzheimer's disease reaches different regions of the brain early. How quickly the disease kills cells in these regions, through the production of toxic protein clusters, limits how quickly the disease progresses overall. The researchers used post-mortem brain samples from Alzheimer's patients, as well as PET scans from living patients, who ranged from those with mild cognitive impairment to those with late-stage Alzheimer's disease, to track the aggregation of tau, one of two key proteins implicated in the condition. In Alzheimer's disease, tau and another protein called amyloid beta build up into tangles and plaques, known collectively as aggregates, causing brain cells to die and the brain to shrink. This results in memory loss, personality changes and difficulty carrying out daily functions. By combining 5 different datasets and applying them to the same mathematical model, the researchers observed that the mechanism controlling the rate of progression in Alzheimer's disease is the replication of aggregates in individual regions of the brain, and not the spread of aggregates from one region to another. The results, reported in the journal Science Advances, open up new ways of understanding the progress of Alzheimer's and other neurodegenerative diseases, and new ways that future treatments might be developed. For many years, the processes within the brain which result in Alzheimer's disease have been described using terms like cascade and chain reaction. It is a difficult disease to study, since it develops over decades, and a definitive diagnosis can only be given after examining samples of brain tissue after death. For years, researchers have relied largely on animal models to study the disease. Results from mice suggested that Alzheimer's disease spreads quickly, as the toxic protein clusters colonize different parts of the brain. The thinking had been that Alzheimer's develops in a way that's similar to many cancers. The aggregates form in one region and then spread through the brain, said Dr. Georg Miesel from Cambridge's Yusuf Department of Chemistry, the paper's first author. But instead, we found that when Alzheimer's starts there are already aggregates in multiple regions of the brain, and so trying to stop the spread between regions will do little to slow the disease. This is the first time that human data has been used to track which processes control the development of Alzheimer's disease over time. It was made possible in part by the chemical kinetics approach developed at Cambridge over the last decade, which allows the processes of aggregation and spread in the brain to be modeled, as well as advances in PET scanning and improvements in the sensitivity of other brain measurements. This research shows the value of working with human data instead of imperfect animal models, said co-senior author Professor Tuomas Knowles, also from the Department of Chemistry. It's exciting to see the progress in this field. 15 years ago, the basic molecular mechanisms were determined for simple systems in a test tube by us and others, but now we are able to study this process at the molecular level in real patients, which is an important step to one day developing treatments. The researchers found that the replication of tau aggregates is surprisingly slow, taking up to 5 years. Neurons are surprisingly good at stopping aggregates from forming, but we need to find ways to make them even better if we're going to develop an effective treatment, said co-senior author Professor Sir David Klennerman from the UK Dementia Research Institute at the University of Cambridge. It's fascinating how biology has evolved to stop the aggregation of proteins. The researchers say their methodology could be used to help the development of treatments for Alzheimer's disease, which affects an estimated 44 million people worldwide, by targeting the most important processes that occur when humans develop the disease. In addition, the methodology could be applied to other neurodegenerative diseases. such as Parkinson's disease. The key discovery is that stopping the replication of aggregates rather than their propagation is going to be more effective at the stages of the disease that we studied, said Knowles. The researchers are now planning to look at the earlier processes in the development of the disease and extend the studies to other diseases such as frontal temporal dementia, traumatic brain injury, and progressive supranuclear palsy, where tau aggregates are also formed during disease. The study is a collaboration between researchers at the UK Dementia Research Institute, the University of Cambridge, and Harvard Medical School. Funding is acknowledged from Sydney Sussex College, Cambridge, the European Research Council, the Royal Society, JPB Foundation, the Rainwater Foundation, the NI, and the NIHR Cambridge Biomedical Research Centre, which supports the Cambridge Brain Bank. And that is it, guys. This is the scientists identify the cause of Alzheimer's progression in the brain. Hope you all like this video, and please don't forget to smash that like button. And you're new to this channel, please hit that subscribe button and also hit that notification bell so you will be updated on my next video upload. Mega love shout out to my premier team, Team Hainan and Team Tulungen, and all its very active and supportive members. This is why the fact not see you all on my next video. Thank you for watching.